Thank you. So thank you for the beautiful introduction, and thank you for the organizers to invite me for the meeting. Of course, I would have preferred to be presentially there, but this has to wait a bit more. So the topic that I will be talking is about endometrial microbiome in health and disease. How can I change the slide now? Yes. So when we talk about microbiome or microbiota, there is always a confusion. What is what? What is human microbiome? What is microbiota? We all have microbes. We think about principally bacteria. But when we talk about microbiome, they are the genetic material of these microbes that live in or on our body. And it's not only, we don't talk only about bacteria, although majority of the microbes are bacteria, but it also includes viruses, archaea, fungi, and other small microorganisms. And the microbiota is like the whole pool of the microbes, and microbiome is the genetic information of these microbes. I have here a slide about terminology, just to make it clearer that microbiota is a community of microorganisms present in a defined environment, and microbiome is include genetic material genomes of these microbes. And I would stress again, it's not only bacteria, it's also viruses, fungi, archaea, and microeukaryota. And when you are not sure, is it correct to say microbiome or microbiota, you can go in a safer way and say microbes or microenvironment or microorganisms. So that is not to create confusion. So we all have our personal genome, which we know already for a long time, but we also have our personal microbiome. So what makes it complex, all the research in the field, is that when we talk about the genome, this is as simple, no? We are 99% the humans are similar between each other, and you inherit half of the father, half of the mother, and is something we understand well. And we have around 25,000, 20,000 different genes in our genome. But then when we go to the microbiome, this is, everybody has the personal microbiome, and we are only 5% similar between people. And although if we talk about cells in our body, the same number, very similar number of cells, our own cells, and bacteria or microbes, we have the similar number of the cells and microbes. But then when we go to the genes, in human we have 25,000 genes, and in microbes we have 3 million genes. It's 100 times more genes. So clearly it's a complex microbiome, microbiota, and we all have our own. We are very little similar between individuals, and considering the amount of genes, it's even called the second genome. Clearly it has impact on our health. So this again to show, it was one beautiful study published a couple of years ago, and they just show here, it's a healthy woman, and they analyze different sites of the body, oral cavity, skin, vagina, intestine, esophagus, and the same samples throughout the body, and what they see is that they are very little similar between individuals. It's just the colors represent different bacteria detected, and both are healthy, and both everything is working well. So that is adding the complexity into the field. So it is very hard when we talk about microbiome analysis that what is bad, what is good. It was rather, I would suggest we take the concept that we have eubiosis and dysbiosis. So it is homeostasis, it's a balance, it's well balanced or out of balance dysbiosis. And then when you think that you are totally alone, you are never alone. So because as I said, we have thousands, millions of microbes in and out in our body. So why is it now so hot topic, the microbiome? Why is it, you hear it even in the announcements, in advertisements in TV, there is a creams that they say microbiome. So basically it is now everywhere, it's entering every field, is somehow touching the microbiome. So it's the boost started with a big human microbiome project in 2007. It was financed from NIH from USA, and the technological advances allowed to go more deeper into the analysis, understanding what is the importance of microbes in our health and in disease. And the first studies of this microbiome project were highlighting that areas that we were thinking to be not so crowded by microbes are rather crowded. So a big part in our field, in the reproductive field, a big part, almost 10% of the microbes they detected belong to the urogenital tract. And the field in the reproductive sciences has been a bit lagging behind the other fields, but it's coming now more and more. So the research in the field of endometrial uterine microbiome has been 5-6 years. And this was a big, beautiful review published 6 years ago already. And it was like the first hint or suggestion that microbes are involved in all aspects of the reproduction, starting from the gametogenesis, fertilization, implantation, and pregnancy outcomes. So if we go to the endometrium, what we know today, the endometrial microbiome, we have published a beautiful review where we have summarized different endometrial pathologies. Here is healthy, infertile, endometriosis, chronic endometritis group, endometrial polyps, dysfunctional menstrual bleeding, endometrial cancer. So basically everything possible to be studied is being studied. And here are the number of references behind which studies have been involved and what are the most common bacteria found in association with these pathologies. So if you are looking any of these boxes, you can see that it's different names. And the truth is, or the sad truth is, that these different studies have different results. It's rather heterogeneous, and we don't have how to say, uniform results for our questions. So in this review, we also highlight why is it so complex? Why do we get so different results? Is because it is everything complex. These are just an illustration of different factors that can influence the results or the interpretation of the investigation. So starting from the BMI, nutrition, ethnicity, how you are sleeping, are you doing any exercise, stress level, all kinds of factors can influence microbial composition in our body. So what we have come out next is that we have proposed recommendations in order to perform well-designed research in this field. There are recommendations for study design, selection of population, endometrial sampling, because this all influences how you are taking the sample, storage, detection of the microbiome, data analysis, validation, and presentation. So all of these steps are crucial. And this is a list of recommendations with the aim to unify different studies in the field so that we could later compare and increase the power in interpretation of the results. And now I would like to highlight key papers in the field that are kind of leading the way in this endometrial microbiome studies. So this is one of the first studies, one of the biggest study performed in more than 100 women. And this figure, I have shown it a lot, and I will show it a lot because it's just so beautiful, illustrative. But what it shows clearly is that when we analyze the reproductive tract for microbes, and the colors represent different bacteria represented, detected. So we can see that in the lower reproductive tract, it's the majority or the composition is composed of lactobacilli. Lactobacillus is almost 100% in lower, and if we go to the cervix, it's 97%. And in the uterus, the lactobacillus is 30%, and the 70% is something else. And if we go to the fallopian tubes, or more the how to say Pau-Douglas, in this case, you see it's even more colorful. So it looks like it's more rich in the different bacteria. The upper you go in the reproductive tract, and this illustration clearly shows the complexity in our research, because if we want to take a sample inside the uterus, depending how we take it, we might influence the results. So the normal way, or the most convenient way of sampling endometrium is through the vagina and cervical channel. So it is very hard to avoid contamination from lactobacilli from the lower reproductive tract. So this was the first study to demonstrate so nicely. And this is another study from the Carlos Simon group that I think boosted in the reproductive or infertility field the importance of wish to analyze endometrial microbiome. What they did was they analyzed 30-35 women, and based on the endometrial microbial composition, they grouped them. They could see the results outcome of the IVF treatment. So in this, the blue represents the lactobacillus. So women who have dominance in lactobacillus, they got the live birth out after the treatment. I mean, one outlier, but we talk in general. This group that was more colorful, they didn't reach pregnancy. And this group, even more colorful, of different colors, there was a group of miscarriage. So this was the first study to demonstrate that their microbiome composition could be somehow linked to the IVF outcomes, treatment outcomes. And this is another from the same group. It's the biggest, it's a randomized trial and multi-center in more than 300 patients. And they see basically the same. They see that in the group of lactobacillus, these are the lactobacillus dominants, and these are other taxa, other bacteria. These are also lactobacillus dominants compared to other taxa. And what we can see here is that they analyze both endometrial fluid and endometrial biopsy. And the results are very similar. So the women who have the lactobacillus dominance in the samples in the endometrial fluid or endometrial biopsy, they were more highly probable to receive live birth after the treatment. And here is a small group with Lactobacillus dominans who didn't achieve the pregnancy. And in the groups where other taxa were predominant and not Lactobacillus, they they associated to the group of women who had biochemical pregnancy and clinical miscarriages in both sample types, endometrial fluid and endometrial biopsy. So the message of the study of more than 300 infertile women is that endometrial microbiome composition before embryo transfer is a useful biomarker to predict the outcome. And this is another study I would like to highlight. I think very important in the field is that they studied 20 women, but the patients were hysterectomy patients. So the endometrial samples were not taken transvaginally. They were taken through the operation, not touching the vagina or cervix. And in these women, of these 20 women, they didn't detect any Lactobacillus dominans. They even, if you see here the line Lactobacillus, you can see here that in endometrial samples, there was no Lactobacillus detected. And there were other groups. And they also confirmed the same in the PCR, focusing only on the specific sequences of the Lactobacillus bacteria. And they can see here that in the vagina, the women had Lactobacillus inners, in the cervix, also inners, and in the endometrium, they couldn't detect the Lactobacillus inners, and Lactobacillus crispatus, that is also very prevalent in vagina, and cervix less here, and endometrium, they didn't detect either. So that is the question, what are we detecting? Are we detecting Lactobacillus strains contaminated from the vagina and cervix or not? So this is actually still an open question. And a very recent study, a couple of months ago, published by Chinese group, and what they show is what we actually expect them to see, but nobody has shown it before. So first, the study was big, had a lot of power, and what they studied was uterine samples, endometrial samples from different age groups. And of course, we expect that with the age, the microbial composition changes, but this study demonstrates it is true. So here you can see the different colors represent different bacteria groups, and you can see that with the age, there are some fluctuations, and with the menopause, bigger fluctuations in the composition. But they also did the study is, I think, very interesting, because what we don't know, where do the bacteria come to the endometrium, not to the uterus. It could be from the blood flow, it could be from previous or sexual intercourse, but also that from the vagina, there is, how to say, connection, no, or interaction with the vagina. And what they did experiment in rats, and they were taking samples from the uterus, from group 1 and group 2. And they did, before they took the samples and after, exchanging the vaginal microbiota from one group with another group. So they analyzed the uterine samples before the vaginal exchange and after. So if somehow the vaginal microbes could interact or go through the channel into the uterus, we should see some influence, we should see some changes. And indeed, we did see, or they did see. So this is the before exchanging. This is the pattern of the rats. and this is after exchanging the vaginal microbes. This is what is happening in the uterus. So there definitely seems to be some communication interaction between these two connected places. And what they also did, they analyzed they had 4 groups of women, control women, or blank control, let's say 3 groups of women, healthy women, women with endometritis, and women with vaginosis. And what they did, they collected the vaginal samples from these women and transfected the rats from the human samples from the 3 different study groups, and sampled uterine endometrial samples to see what is changing. Does it lead to any differences between healthy women, endometritis, vaginosis, if we do the exchange or not exchange, if we do the transfection from the microbes from samples from the vagina into rat vagina, do we see any influence inside the uterus? And they did see. So they analyzed different inflammatory markers, and what they saw is, if we compare the blank controls or healthy, there were no blue and green, there is no much differences, but the women with endometritis, the biggest, and also vaginosis, influenced the intrauterine environment of the rats. So there clearly is an active interaction between these 2 sites in the microbial composition. So to finalize the talk, I would like to change a bit and highlight, or how to say, stress. Until now, what I've been talking is microbial analysis and analysis on the level of DNA. What we've been analyzing here is the 16S RNA gene sequence. So every bacteria has ribosomic genes, and based on the sequence, we can detect which, because they are very unique for every bacteria, we can detect which bacteria we have present in the sample based on the sequences of DNA. But then there is another option. You can analyze on RNA level. You can analyze RNA sequences, bacterial RNA sequences in the sample, and what would you get? You can detect also the presence of bacteria or viruses or fungi or other microorganisms. What is the difference if you detect DNA or if you detect RNA? If you detect DNA, DNA is a very stable molecule, so it can even be alive, not alive. The sequence can survive thousands of years. So if you have had a infection, for example, and the bacteria is treated or eliminated, the sequences still remain. So when we're analyzing, we are talking about the microbiome studies, we actually don't know if the bacteria or microbes we are detecting are alive, or they are just some leftovers or some just degradation sequences. So we are kind of guessing that when we detect this DNA sequence, the bacteria exists or the microbe exists, but we don't know it. So, but when we analyze the RNA, RNA is a very unstable molecule. So if you can detect any RNA sequence in a sample, it means there is some activity, because no dead cell is producing RNA molecules. RNA molecules are only produced when you need a protein of this. So if you detect sequences of RNA, you can be assured that there is active bacteria producing or viruses producing these sequences. There is also, you can analyze protein level or metabolite level, but this is more complex. This will be the future. We are not there yet today. So what we did until now, all the studies have been analyzing DNA. 16S or whole genome analysis. There are couple of studies done today from the endometrium because it's a low microbial biosite and it's not easy to amplify the whole genome of the microbes. So most studies, 99% studies have been analyzing 16S RNA gene sequence. So what we did, we mapped the entire functionally active endometrial microbiota. We analyzed the RNA sequences of microbes. So here I cannot show, unfortunately, but it's also inside this publication. You can see there is a link. This is the link, and this is an interactive map. So we created based on the RNA sequences, based on the functionally active microbes. If you are in the site, you can click here, and it will jump like bigger, like for example, here you have bacteria, you click here and comes all the scope of all the different bacteria you can see. There are viruses, and if you can click to the place of the viruses, you can see which viruses were detected and so on. So it is a very beautiful tool. It's like a toy to play with around. And what we also did, we analyzed the samples. First, we detected more than 5,000 different functionally active microbes in human endometrium, only healthy. In this time, we analyzed only healthy. And we compared proliferative phase and secretory phase, just to see if the menstrual cycle has any difference, does influence the composition. And we saw that it does influence. So there are not big differences, but small differences, slight differences between the before the ovulation and after the ovulation. In the proliferative or in the secretory phase, there are differences in the microbial composition, the active microbial composition. And we did even a step further. We were predicting based on the sequences, predicting which kind of pathways could be interact, because what we are doing so far is we are detecting the microbes on DNA or RNA level, but we have no idea what are they doing, what is the influence in the uterus. Clearly they must have some influence, otherwise they wouldn't be there, but we don't know what. So this is kind of first step to model, it's in silico analysis, in which kind of interactions, host microbe interactions could be involved. And we analyzed with the blue, you can see pathways that we were detecting in the endometrial samples in human, in different melatonin, in serotonin and different pathways, and tryptophan. And the red one are the pathways that we detected in both microbes and in humans to be activated. So what we see is that in the serotonin degradation pathway, there seems to be activity on both sides. So we could expect there is a communication interaction between, and the tryptophan degradation pathway and also prostanoid biosynthesis pathway. So this is kind of first hint to show that there are more to this than just detecting the sequence of bacteria. So what I would like to conclude, it's just so beautiful figure that I wanted to show it, that it is rather colorful, the picture inside in the endometrium, and we are still steps away to understand what is the function, how are they influencing. So I would end up with my presentation with more questions than answers. Knowledge of this, sorry, there is a mistake. Knowledge of the microbe composition in all reproductive tract is in its infancy. The core microbial composition in health and disease needs still to be established and confirmed in many studies. We don't know what we are talking. What does it matter actually, DNA sequence or a live bacteria? Are these microbes that we are detecting residents or tourists or invaders? Is the dysbiosis that we are seeing in different pathologies, is it the cause or the consequence? And can the dysbiosis be treated? If it were treated, we should expect that these bacteria are alive. So these are more questions than answers. And I'm expecting some questions from audience. Hopefully I can answer. And the last slide is to thank the research team and the funding bodies in our research. Thank you very much.
Thank you very much, Dr. Altmae, for a very interesting lecture. So the question about microbiome is very important, as we saw from your lecture. You say that treatment could be help for a patient, could be help for our result in ART treatment. What kind of treatment? You say antibiotics, but do you know any other type of therapy which could be performed in this patient? This is my first question. And the second one, in which patient we should perform NGS as analysis for microbiome in women, and what kind of sample you can recommend, fluid or biopsy?
Okay, I will try to answer. So which kind of treatment? What I was hoping to give with a message with the talk is that I think we are it's too early to treat yet based on the microbial composition, because even if we see, okay, we have Lactobacillus dominans or we don't have Lactobacillus dominans, that doesn't mean that these bacteria are alive. So when you are treating with antibiotics, you are treating a live bacteria, you are killing a live bacteria. So if we are only analyzing DNA sequences, we cannot make sure what are we killing there. We are killing maybe that we should not be killing. So I think we are a bit step behind. I mean, I understand there is always, where is research and where is the demand from the clinics and the doctors. So we are not in the same level. The research is behind from the demand. And I know clinics where they are treating, they are doing the test and they see that it's not Lactobacillus dominans and they are treating with antibiotics and then probiotics. There are different companies offering probiotic solutions, oral pills or vaginal, and but we cannot say if it's correct yet, or is it helping? Even we don't know exactly what is it we are treating. So me as a scientist, I would say it is too early to start to treat with antibiotics. Clearly, if you have pathogens, you have to go for antibiotics. But just based on the microbiome analysis, in my opinion, today it's too early to treat with antibiotics. So this is the first question.
The second was for the samples. Which one do you recommend personally, fluid or biopsy for NGS?
Yes, I personally recommend biopsy because it's just so much more informative as a scientifically talking. No, patient side talking, of course, clearly fluid is less invasive, less painful. There is so much less studies done in the fluid, so I cannot say that it is identical. Today, I know that when we are talking about the bacteria or viruses or the microbes, the uterus is a low biomass site, so there is not so many. So if we get the biopsy, we get more information. We ensure that we get enough sample for analysis. So in that way, I would recommend the biopsy. But of course, I think fluid would be the future. It would be best if we could analyze only fluid and just before the procedure, so before the embryo transfer. That will be less invasive, and we would get the answer. And then what else? Which samples and what to analyze? What would be the future? I would even expect if we could maybe just analyze the cervix. We can take a sample of the cervix and we could get some answers, some hints. What could be the environment inside the endometrium? That would be the best. That is the less invasive and the fastest.
Okay, good clue. Another question, Professor Kirchev, please.
Thank you for the very interesting result about the RNA. But have you tried to isolate living micro bacteria from there, parallel to your studies using the RNA sequencing and so on? Have you tried to isolate living bacteria from these healthy persons?
No, we haven't done it yet. This is something.
Sorry, has anyone showed that in healthy endometrium there is a bacteria which can be isolated by microbiological methods?
The thing is, with the microbiotic methods, we are going for DNA RNA analysis. So it has to be then the culturing method, the traditional culturing. And we know with the culturing methods, we can grow less than 1% bacteria, what we are detecting inside. So the problem is also that inside the uterus, there is, how to say, the environment. We actually even don't know the exact pH. We know it's around 7, 8, but different areas have different conditions. So there are they are anaerobic. So it's not very easy to grow this. I think I haven't seen anybody doing it, because technologically it's very hard.
So we have to believe only with molecular biology methods and technologies for showing the microbes within the uterus. Is that right?
Today, yes. But for vagina, could be done the culturing.
Well, I'm not asking about vagina, I'm asking about the uterus. Thank you very much.
Thank you. Any other questions? Yes, Dr. Kudukcheva.
Thank you so much for the interesting lecture. I have a question. You said there's difference between in the menstrual period when you take the sample before or either after the ovulation. What did you mean are these differences? And by your practice, which one is better, to take the sample before or after the ovulation?
The differences were small but significant. And for your practice, I know it's easier to take in the proliferative phase. I don't know this question or answer to this question, because it's what do you want to see? I am, as a scientist, I'm interested what's happening in the implantation window. So that's why I wanted to compare with some other situation to see what is happening in the implantation window. But if to go for, let's say, biomarkers in the future to predict the environment. So what we want to see finally, what you want to predict the environment is you want to see if the embryo would implant or not. You want to see actually what is important. Doesn't matter what is the environment in the proliferative phase for you in your setting. So then I would go for the secretory phase, yes.
Thank you.
Thank you. Any other questions from the audience? No. So one short from Professor Timeva.
Thank you very much about this interesting result you present. As you mentioned, you are a scientist, but from the practical point of view, what is your recommendation? Is it necessary to give antibiotic after oocyte retrieval in every patient? Because you know that it's invasive procedure. And after a few days, we make embryo transfer, and the catheter of embryo transfer pass through the cervix and may introduce some kind of bacteria of endometrium. Because you know that a lot of infertility units have a regular antibiotic prophylaxis, let's say, after oocyte retrieval. From your point of view, what is your recommendation? Thank you.
It's a very hard question for me. From my point of view, it would not be needed, the treatment, but you don't want to run the risk of infection. So as a clinician, you always have to, one thing, think of what is reasonable, but what is best for the patient. So I don't know what is the prevalence of the infections after, because as a routine, you give the treatment. So, and you don't want to make a study to do a control group. We don't give you the treatment and maybe you get the infection. So I think in the current situation, it's more important to avoid the infection than to treat or to do the antibiotic treatment. I would hope that there will be some local products like capsules or that could maybe prevent infections. If we are there, then we can avoid the antibiotic treatment. Or probiotic, if you do the treatment, you could, how to say, aid or help the environment not to be so killed by the antibiotics.
Okay, thank you.
Okay, thank you. Let's finish this topic and continue with the next speaker. Thank you, Dr.
Thank you.
Altmae. Bye.